Okay, so now we're just going to finish up. We're going to talk a little bit about proximate mechanisms and ultimate goals, and a little bit about the EEA, the Environment of Evolutionary Adaptation, and then we're going to talk about hormones, and I think you'll see how these things kind of roll together. It's pretty cool. Okay, so when we're talking about proximate mechanisms versus ultimate goals, what I want you to have in mind is not just how this behavior it works right now, okay? I do want you to talk about that, but also how a behavior would have served our survival and reproduction in the environment of evolutionary adaptation. So let's say that I asked you, for example, about jealousy. You know, why would we have evolved to have jealousy? Well, the proximate mechanisms for things like jealousy are in a situation, you might feel that another person is spending too much time with your significant other. That's a perfectly reasonable explanation for why you feel jealous. Or you might talk about social psychology and say, you know, these are the social factors that contribute to jealousy. Or you could say developmentally, maybe hormones contribute to those feelings of jealousy. So anything that you're talking about an explanation for here and now. But then you want to also be thinking about, in the environment of evolutionary adaptation, the environment in which human beings evolved, the environment in which any organism evolved, how did that behavior help them survive and reproduce? Well, if you were jealous of your mate, say for example, if you were a male, that might keep your female mate from reproducing with another male. Or if you were a female, it might keep your male from giving male parental investment to another female. So feeling jealous may have served our survival and reproduction in that way. Okay, so that's what I want you to be thinking about. Because what happened in the environment of evolutionary adaptation, what happened in the EEA, the pressures that were around in the EEA are not necessarily the same as you find today. So even though jealousy may have helped us survive and reproduce in the EEA, is it really helpful for us to feel it in the modern day? You know, is it really helpful to have those feelings? And if not, what should we do with them? Those are the kinds of questions I want you to be thinking about. Okay, so now let's talk about hormones. And there's a big overall concept that I need to explain, and then there are a few technical details that I'm also going to talk about, and then it's going to eventually get to this really cool point. And so if you can bear with me and follow along through all of the different steps, number one, there's going to be an essay question about it on the test, I can guarantee you that. But number two, I think you're going to think this is really, really cool. It's one of my favorite things to talk about during lecture. Okay, so the big main takeaway concept here is about the difference between activating and organizing effects of hormones. Okay, and that's what I'm going to talk about first. What hormones are and what those two different kinds of effects are. Okay, so what are hormones? Well, hormones are chemicals that are secreted by glands in our body. Could be the pituitary gland in the brain. It could be, your adrenal glands down on top of your kidneys. So you have glands that secrete hormones throughout your body and brain, and those glands are called endocrine glands. And what hormones do is they travel through the bloodstream, and then there are cells in various organs in our body that those specific hormones interact with. In the same way that a neurotransmitter would cross the synapse and then interact with the postsynaptic cell, hormones go through your bloodstream and then interact with various cells. And I'm going to talk about a couple of the different ways that happens. But we call those cells a target cell, so a hormone has a target cell, and we call the organ that those cells are part of the target organ. So for example, if adrenaline is released into your body and it binds to cells on your heart is the target organ, and your heart cells are the target cells, for example, okay? So these are the different kinds of effects that hormones can have on the body, and this is going to be the main gist of what I'm talking about here. When most people think about hormones, they think about the activating effects of hormones. So for example, your hormones come and go throughout your menstrual cycle, and they cause you to have your period, and they cause you to ovulate, and all of those good things that happen that are associated with the menstrual cycle. Those are activating effects of hormones. Or if you have hormones coursing through your body and you have certain feelings, so sometimes when people take certain hormones, they have certain emotional experiences. Those are the activating effects of hormones. They are things that come and go. So for example, when you get pregnant, a lot of things have to happen in your body to support that developing fetus, and many of those things, many of those processes are triggered by hormones. So for example, first you need to thicken up your uterine wall, then you need to build a placenta, then you need to loosen up your bones so that you can pass a baby out through your pelvis. You need to increase the volume of your blood. You need to do a lot of different things during pregnancy. And then once you have the baby, you bond with the baby and you nurse the baby. And all of those things occur in part because of the activating effects of our hormones. We can do those things because our hormones come and go according to our needs at the time. However, there's another kind of effect that hormones have, and that's called an organizing effect. And this is an effect where once it happens to your body, it changes the body in some kind of permanent way that you can't go back from. So for example, hormones are involved in growth. But once you grow, you don't shrink and grow and shrink and grow and shrink and grow as your hormones come and go. Once you grow, you're that height pretty much for the rest of your life unless something unusual happens. When you start to produce hair during puberty, like men get a beard and everybody gets armpit hair and pubic hair, and then you have it for the rest of your life. Unless something unusual happens, you have it for the rest of your life. When you go through puberty, men's voice deepens, women grow breasts and hips and all of that good stuff. And then once it happens, it's that way forever. Those are organizing effects. But I'm going to come back to this in a really important way, so that's the first thing you need to know. Okay, now I'm going to talk about a few different kinds of hormones and the differences between them. First, I'm going to talk about the amine hormones. And you know some of the amine hormones. Up there you see norepinephrine and epinephrine. And you know norepinephrine is one of the neurotransmitters we've been talking about. And amine hormones also include other neurotransmitters like melatonin or thyroid hormones, hormones that are secreted by your thyroid gland. And they work essentially in a very similar way to a neurotransmitter. So yes, for example, norepinephrine is something created by your brain and released across synapses, but it's also secreted into your bloodstream and circulates through your entire circulatory system, and then it will act on receptors on different cells, on different target organisms. Or target organisms, Teresa? Target organs. Sorry, you guys. Target organs. Target cells and target organs. So that's what amine hormones do. They go through your blood, they travel to whatever the target organ is, they go to that target cell, and then there's a receptor on the cell, just like there's a receptor for neurotransmitters on neurons, except for this is on your other body cells. And the hormone docks to that binding site and then has an action on the cell. That's amine hormones. Protein hormones are very similar. So those include guys like insulin and growth hormones, FSH, LH, these kinds of different hormones. And they also interact with receptors on target cells, on target organs. However, they're called protein hormones because they trigger the cell to use protein, to utilize protein. So for example, when you're growing, you need to tell your cells, okay, build more, get bigger. And so that kind of message triggers various proteins to be utilized in the cell to help you grow. okay? Simple enough. But then the third kind are called steroid hormones, and those include some of your sex hormones. So for example, your androgens. The famous androgen you've probably heard of is testosterone. And then other sex hormones like estrogen and progestin, okay, the big famous sex hormones, and also your glucocorticoids, those are in the same category. But the reason that steroid hormones are so powerful is because they cross directly through the cell membrane and actually interact with the DNA itself. Okay, so they don't have to bind to a receptor. They can cross right through the cell membrane, right into the nucleus, and there's actually a receptor for that hormone inside the DNA, and then it directly turns the DNA on or off, depending on what needs to be done. And so these hormones are involved, obviously, in our sexual behaviors and our reproductive behaviors. And then some of these steroids, for example, testosterone, are abused by some people because, you know, testosterone is something that our body naturally releases when we are building muscles. And so people who want to build muscles bigger and faster will sometimes inject hormones like testosterone in order to try to bulk up faster than you can naturally. And of course, that's not healthy for our bodies. But there's not all sex hormones are steroids. The ones that we just talked about, your testosterone, your estrogen, your progestin, those are steroids. But there are other sex hormones that are actually in the protein hormone group. And we call these guys gonadotropins, okay? Tropin means movement or activity, and your gonads are your testes or your ovaries. So it causes action of the gonads. Okay, so those, for example, FSH and LH are two gonadotropins, two sex hormones that are actually in the protein hormone category, and they are responsible for like regulating your menstrual cycle when you ovulate and when you menstruate and so on and so forth. Okay, so here's the thing about these hormones. They have activating effects, yes, and I'm sure you're familiar with people talking about the activating effects of hormones, but they also have organizing effects that are really super important to understand for the purposes of this lecture. And yes, there's going to be a question on this on the test. Okay, so I'm sure you remember that every human in every cell of our body has 22 autosomes, 22 pairs of autosomes, 22 pairs of autosomes, 44 total autosomes, and 1 pair of sex chromosomes. And for females, you remember what the sex chromosomes are? X and X, does that ring a bell? And for males, usually the sex chromosomes are X and Y. Hopefully that rings a bell. Now, of course, there are people who have other genes. You can have 3 X's, you can have 2 X's and a Y, an X and 2 Y's. But typically, most females have 2 X's and most males have 2 Y's. And the thing that's really, really, really important about the Y chromosome is that it has a certain gene on it called the SRY gene. And the SRY gene is extremely important because it is responsible for making a male a male. So in a typically developing male, the Y chromosome has the SRY gene, and the SRY gene, the first instructions that this gene is going to send are for making testes. Okay? So testes are created by the SRY gene. It turns on parts of your DNA that says make testes. And then once you make testes, guess what testes make? Testosterone. The hormone testosterone is called testosterone because it's made by the testes, okay? And so then the testes are going to crank out testosterone throughout development, both in utero and after you're born. Now, everybody has all of these hormones in their body. Everybody has testosterone, estrogen, progestin in their body. It's just that males have more testosterone. Okay, so here's normally what happens when you have testosterone in your bloodstream. Testosterone, like I say, is made by the testes, and because it is a steroid hormone, it crosses right through the cell membrane and goes directly into the nucleus of the cell, and it interacts with the DNA in the nucleus of the cell, and what it does is it masculinizes the cell. So for example, let's say you're trying to build a penis and you're trying to build all of the things that go along with the male anatomy, the vas deferens and the scrotum and all of those different parts of the male anatomy. Testosterone crosses through the cell membrane into the cell, activates the DNA, and says, hey DNA, you're going to become a penis cell or a vas deferens cell or whatever the cell is. It masculinizes the cell so that it's going to become whatever it is. Now here's something interesting. When testosterone enters the cell, it actually gets converted into estrogen inside the cell. And so it's actually estrogen that masculinizes the cell. But here's the thing about estrogen. Yes, estrogen is around the cell during development. But we also have another chemical in our bloodstreams called alpha-fetoprotein, AFP. And AFP is the thing that they measure when you go into the doctor and they tell you how far along you are in your pregnancy by how much AFP you have in your bloodstream. And what the AFP does is it binds to estrogen. So you have AFP circulating around your bloodstream and it binds to estrogen. And so estrogen, because it's bound to alpha-fetoprotein, is too big to cross the cell membrane and too big to enter the cell and masculinize it. However, alpha-fetoprotein does not bind to testosterone. Testosterone tricks the alpha-fetoprotein so that it gets to sneak in to the cell, get converted into estrogen, and masculinize the cell. In fact, if you gave the person too much estrogen, and so there wasn't enough alpha-fetoprotein in their bloodstream to bind to all the estrogen, estrogen would actually cross into the cell and do the exact same thing as testosterone. So estrogen and testosterone actually both would have masculinizing effects on the cell, except for the fact that alpha-fetoprotein binds to the estrogen and keeps it from entering the cell when the baby is in utero. Okay. So here's the deal. If you have the SRY gene, usually it is going to masculinize the genitalia. When you are first in utero, okay, when a human is first conceived, and all the way up through 6 weeks, the genitals of the fetus are undifferentiated, meaning they could either become male or female. In fact, female is the default gender. If there is no instructions from the SRY gene, if there are no instructions from hormones, then the fetus is going to develop along the female line. You're going to see by 10 weeks, the genitals are starting to change and take on the look of female genitalia. And by the time you're 20 weeks pregnant, this stuff is all done. Okay, this stuff is all done being constructed. They can give you an ultrasound and they can show you the genitals of the baby, and then that's what they have for the rest of their lives. Whereas, if you are a male fetus, the SRY gene is going to create the testes. The testes are going to make testosterone. Testosterone is going to circulate through the baby's bloodstream, and it's going to masculinize the genitals. And so the testosterone will start to instruct the cells, build a penis, build a scrotum, build vas deferens, a prostate, all of the stuff that male genitals have that make them male. And then by the time that fetus is 20 weeks along, that's all set in stone. So the critical period of development for the genitals is around 6 to, definitely less than 26 to 16 weeks in utero. Okay? So once that happens, that's the genitals you have forever. Okay? So if you have an SRY gene, and if the SRY gene functions typically, it will make testes. The testes will start to pump out testosterone, and then that will masculinize the genitalia during the critical period of development for the genitalia. Okay, so if you have your whole time in utero, early 6 weeks, 10 weeks, okay, around that time is the critical period of development for the genitals. After that point, no matter what genitals you have, those are the genitals you have forever. No matter what hormones you get after that point, it won't change your genitals. So it should be fairly easy to see how you get an intersexed individual. So the person could have been developing along the male line and then stopped at a certain point. Or they could have been developing along the female line and then at a certain point started to get instructions to create male genitalia. And so if any of these processes were halted anywhere along the way or changed anywhere along the way, you might get genitals that are neither male nor female. Okay? So the female's the default gender. The female is the way that they will behave in the absence of any other, the way that they'll be formed in the absence of any other hormones. Okay? So female is the default gender. Sometimes people think, oh, male first and then females are a take on the male. It's exactly the opposite. Females are the original and males are a take on us. Okay, so SRY gene creates testes, masculinizes the genitals during the early part in utero. But here's the thing. There's also a critical period of development for the gender of the brain. Male and female humans do have some differences that are detectable in the brain. And one of these things that's quite apparent by asking people is their experience of their gender. Okay? So the critical period of development for the gender of the brain, meaning masculinizing or feminizing the brain, that happens late in development. The critical period for the brain getting its gender happens after the critical period for the genitals. In a typically developing male, the SRY gene instructs the testes to be made, then the testes make testosterone, and testosterone masculinizes the genitals first, and then it keeps going. There keeps being more and more testosterone flowing through the bloodstream of the fetus, and it masculinizes the brain during the critical period of development for the gender of the brain. But those are two different things happening at two different times. And in a typically developing male, you get a masculinized set of genitals, meaning you get a penis and scrotum and all that stuff. And you get a masculinized brain, meaning the person thinks, hey, I'm a male. Or if you don't have the SRY gene and you don't make testes and you don't crank out a ton of testosterone, then your genitals are going to go along the default and become the female genitals. And then after that happens, there's no testosterone or very little testosterone in the bloodstream during the critical period of development for the brain. And so once that's over, then that's over too. And so for females, you get the default genitals, then the default brain, as long as you don't have testosterone pumping through your circulatory system. Okay. So that's the critical thing to know about these organizing effects of hormones. Once these genitals are formed, that's it. After the critical period is over, there's no going back. They are what they are. Once the brain critical period of development is over, then that's it. The brain knows its gender, and there's no going back. All right. So we have used a rodent model in order to understand these processes a little bit better. And the first thing that I want to say is, human beings are much more complicated than, our brains are much more complicated than a rat brain. And therefore our behavior is much more complicated than rat behavior. And in fact, we've done these same experiments at this point with other animals too, like cats and dogs and even monkeys. But no matter which one we used, they don't have brains as complicated as ours. So number one, for humans, it's more complicated than this because we have more complicated brains. And number two, it's more complicated than this because in this experiment that I'm going to talk about, we very clearly manipulated the amount of hormones that the animals were being given. We did an all or nothing kind of manipulation with the hormones. So in reality, in real life, it's not that clear cut. In real life, hormones can come and go throughout the pregnancy, and certainly for people, all during a 9-month pregnancy, you can have different effects of hormones. So that's what I want to say first of all. It's more complicated than this when it comes to humans, but I think you'll see that this model is very helpful for understanding certain issues. Okay. Male rats exhibit a very distinct mounting behavior that they'll show once they reach sexual maturity, and that is their clear-cut male sexual behavior, and you'll see males do this when it's time to mate with a female. Females have a very different behavior that they do, also once they reach sexual maturity, when it's time to reproduce. They exhibit this female behavior, and this is called lordosis. Okay, so I'll write that word down, lordosis. And so in this experiment, what we were trying to do was see whether the rat would exhibit the male behavior or the female behavior. And if they exhibit the male behavior, we can assume it's because they have a male brain. And if they exhibit the female behavior, we can assume it's because they have the female brain. But here's the thing about the critical periods for rats. It's a little bit different for people, because the critical period for the genitals in rats is in utero. But the critical period for masculinizing or feminizing the brain is postnatal. They actually have their brains masculinized or feminized after they're born. Because if you think about little rats, they're born like little fetuses. They don't develop as long in utero. But with a typically developing male, you will see that the testosterone, the testes are created in utero. The testosterone circulates through their system and makes penis and testicles during the critical period of development for the genitals. And then after they're born, the testes keep on working, and so they masculinize the brain in about the first 2 weeks, 10 days to 2 weeks postnatal, after they're born, okay? Whereas if it's a female, no SRY gene, they're getting ovaries instead of testes, and so the genitals are continuing to be feminized during the in utero part, and then when they're born, they're not cranking out a lot of testosterone, so their brain resorts to the default gender, which is female for their brain. And that's typical development for rats. Very similar to people, except for the brain period of development is on the outside, is postnatal, is after they're born. Okay. So, I can, and researchers have, manipulated these things in order to see what happens. And let's talk about that now. First of all, they test all of these animals to make sure that they have the genes that matches their genitals when they're first born, okay? So they look at the male rat, they make sure he has an X and a Y chromosome, and then when he's born, because he had that Y chromosome, the SRY gene created the testes, which created testosterone, and it gave him penis and testicles. So when he's born, we can neuter him, we can castrate him. We remove his testes. And then if we don't give him any additional hormones during those first 2 weeks, he's going to have a penis, but during that time when he would normally be getting masculinized by the testosterone in his brain, he doesn't have that. And so, it's actually a she. This animal, despite the fact that it was born with a penis and testicles, if we remove the testicles and therefore remove the testosterone during the critical period of development for the brain will resort to the default gender and it will be a female brain. And you will see a rat that does lordosis, that does the female behaviors very clearly. But if I take an animal that's a chromosomal male, and he has penis and testicles when he's born, and I leave him with his testicles for those first 2 weeks, but then I neuter him after that point, after the 2 weeks, he's going to exhibit male behaviors. Because now he had the male genitals to begin with. When he was born, he continued to have the male genitals pumping him full of testosterone, masculinizing his brain. And so he will turn out to behave like a male and do mounting behaviors. If I take a male and I neuter him at birth, but then instead of the testosterone that his testes would normally produce, I inject him with testosterone during those first 14 days, during those first 2 weeks, he will turn out to have male behavior. Because even though he didn't have his own testes, I replaced the testosterone by injecting it into him during the critical period of development for his brain. On the other hand, if I have a female rat, okay, so no SRY gene, no testes, no testosterone or very little testosterone, doesn't make testicles, she has the default gender of her genitals. She has ovaries and vagina and all that good stuff. If I take out her ovaries, though, at birth, what happens during the critical period of development for her brain? Well, it's going to be the default gender, and the default gender is female. So she's going to exhibit female behaviors, because the ovaries have nothing to do with this. The ovaries do not produce tremendous amounts of testosterone or tremendous amounts of estrogen that would masculinize the animal's brain. Okay? What if I have a female and I don't give her any kind of surgery, but I fill her full of testosterone for those first 2 weeks during the critical period of development for her brain? What do you think is going to happen? She is not a she, it's a he. So even though this organism has female genitalia, during the critical period of development for her brain, I injected her with testosterone. And so the brain is going to be a male brain, and you're going to see the male behaviors, going to see the mounting behaviors. What if I take that female and I give her, and I spay her, I get rid of her ovaries, an ovarectomy, when she's born, and then I give her testosterone days 1 through 10. Is she going to have male behaviors or female behaviors? Male behaviors. Because again, it has nothing to do with the ovaries. It has to do with the fact that I gave her testosterone during the critical period of development for her brain. His brain is more like it. What if I have a female, a chromosomally female rat, I spay her at birth, I give her an ovarectomy, and then I give her testosterone after the 2 weeks, after the 10 days to 2 weeks is up? How's her behavior going to be, male or female? Female. Because if I don't give her testosterone, she doesn't make testosterone here, and I don't give her testosterone during the first 2 weeks, the critical period of development for her brain is over. This was the critical period of development for her genitals. This was the critical period of development for her brain. She didn't get much testosterone during either, so she's going to show female behaviors. Because it doesn't matter what hormones you give her after the 2 weeks is up. Maybe she'll be a little bit more aggressive with extra testosterone, but it's not going to change her brain in any kind of organizing effect way. And it's certainly not going to change her genitals. The critical period of development is over for those. What if I have a female, I don't spay her or anything, but then I give her extra estrogen on days 1 through 10. I give her extra estrogen during the first 2 weeks. Okay, if you see estradiol, that's another word for estrogen. It's just usually when it's the naturally occurring kind inside you, we call it estrogen. And if it's exogenous, we call it estradiol, but it's the same. It's the same chemical, okay? What if we give her extra estrogen during those first 2 weeks? It's going to do the same thing as the testosterone, and it's actually going to masculinize the brain. Because remember, testosterone gets converted into estrogen inside the cell. And the only reason that estrogen doesn't masculinize a cell is because alpha-fetoprotein binds to it, AFP binds to it, so it can't get into the cell. But if we give you a whole bunch of extra estrogen, more than you have AFP to bind to it, the estrogen will go across the cell membrane and do the same thing as the testosterone would do. Okay? So extra estrogen does the same thing as giving testosterone. What if I have a male, genetically male, I neuter him at birth and I give him extra estrogen during the critical period of development for his brain? How's he going to turn out? What's his behavior going to be like? Male. Because SRY gene during the critical period of development, which is in utero for the genitals, masculinize the genitals so you get penis and testicles. Then I remove the testicles, but I pump him full of extra hormones. I pump him full of extra estrogen. It does the same thing as the testosterone would do. If there's too much of it, not enough AFP to bind to it, it will cross the cell and do the same thing. Okay, so it should be very clear to see the implications that this has for transgender issues. Okay, there's clearly a critical period of development for the genitals that's early on in development, and once that critical period is over, boom, those are the genitals you have. Okay, and they can be male genitals, female genitals, or anywhere in between, depending on the hormonal message. This, with the rats in this experiment, is very clear cut, whether they got typical male or typical female genitals, typical male or typical female brain, because we deliberately manipulated the hormones to be very clear cut. Either you got a ton of testosterone here, or a ton of testosterone here, or both, or neither. Okay? But in regular development, both in animals and in humans, it doesn't work that way. You may have hormone spikes go up and down throughout the pregnancy. Okay? In a typically developing male, it's going to be testosterone the whole way along. In a typically developing female, it's going to be not testosterone all along. So you're going to get the default gender, which is female. But it should be pretty easy to see how these things don't necessarily match up, and how you might get a transgender person or a non-binary person if you got some kind of intermediate hormonal influence. And there's no way we can know that ahead of time. We don't know by looking at a person, what kind of hormones were or were not occurring typically during their pregnancy. And so you see all of these individual differences that are beautiful. But again, I want to make clear that even though at this point we've done this with, like I say, rats, mice, cats, dogs, monkeys even, okay? So monkeys, the critical period for both the brain and the genitals is they're both in utero like it is for humans, because they have, relatively long gestation periods, 4 to 6 months. So that's more similar to humans, but again, even with the monkeys, we either, tons and tons of testosterone during the critical period for development of the genitals, and or tons and tons of testosterone during the critical period of development for the brain, and then we give them testosterone during the critical period of development for the testicle, or for the genitals, but not the brain, or not for the genitals, but yes for the brain. We can manipulate those things with a monkey, but with humans we don't do that. And so you get all these intermediate expressions, and you can be anywhere along on the spectrum in terms of both your genitals and the gender of your brain. And they don't necessarily have to match. Okay. What does that mean for all other kinds of issues that have to do with experiences of gender and experiences of sexuality? We don't know. We're still learning. Okay. See you guys at the final.